The promoter is upstream of the gene. Okay, what does that mean? Hi biology friends, it's Sarah, your other biology professor. Let's talk upstream and downstream. Upstream and downstream are used to talk about where something on DNA or RNA is relative to something else on that molecule. Now, here's an mRNA. Don't stress out if the parts don't look familiar. They're not what's important here. You may remember that strands of nucleic acids, DNA and RNA, have directionality, a 5' end and a 3' end. And if you don't remember that, I've got a video that clears all that up. I'll link it in the description. Upstream means toward the 5' end of the molecule. So the 5' cap is upstream of the start codon. And downstream means towards the 3' end. The stop codon is downstream of the start codon. Making sense? Try this one. The start codon is blank to the stop codon. Upstream, right, the start codon is toward the 5' end of the molecule relative to the stop codon. It is upstream of the stop codon. So with single-stranded nucleic acids, it's easy, but with double-stranded DNA, we've got two 5' ends, and they're in opposite directions. So if the promoter is upstream of the gene, which 5' end are we talking about here? When we're talking about DNA and a gene, when we say upstream, we mean towards the 5' end of the coding strand. Now, the coding strand is the strand of DNA that is not used as the template for transcription. This sets up the promoter to be before the gene when you're talking about the direction of transcription. So RNA polymerase can hop on and do its thing, transcribing the gene. Now, keep in mind, genes can be found on both strands of DNA, so which one's the coding strand is all relative to the particular gene that you are talking about. Maybe for a different gene, the top strand is the template strand. That makes the bottom strand the coding strand, and upstream is to the right. Since the promoter is upstream of the gene, in this picture, would the promoter be on the left or right side? Which way is upstream of the gene? The template strand is the bottom strand here, which makes the top strand the coding strand, the important one for upstream, downstream. And you can see that the 5' end of the coding strand is to the right, so that's where you'd find the promoter for this gene. If upstream and downstream make a little bit more sense and you know where to put a promoter for the gene, drop me a like so that other students can find this as well. In the meantime, subscribe for more biology content, tips, and tricks from me. Check out my website for tutoring resources, and I will see you next time. Bye.